Hello everyone, this video is about Chapter 9, Homeostasis, Subtopic 9.1, Concept of Homeostasis. At the end of the lesson, students should be able to explain the concept of homeostasis and describe the homeostasis control system. Homeostasis is defined as the process of maintaining biologically stable condition inside a living organism through physiological processes. So the variables of the internal environment that need to be kept constant include our body temperature, the pH of the blood, glucose concentration, concentration of carbon dioxide and oxygen in our blood, and also the concentration of ions, for example, sodium ion and potassium ion. All of the toxic substances in our body must be removed totally or at least be kept at low levels. The optimal level in a homeostatic control system is known as the reference point or a set point. The efficiency of the system is measured in terms of the deviation from the set point and how quickly this set point is restored by the system. The importance of homeostasis. Homeostasis will provide a stable internal environment in the organism by minimal unavoidable disturbance. This is to provide suitable environment for the reactions in our body to occur. The homeostasis also acts as a buffer to the body against fluctuation from optimal conditions, where the body functions more efficiently because its constituent cells are maintained at optimal condition. Fluctuation refers to the changes in the body. The components of the homeostasis control system. Homeostasis control system consists of three components. The first one is the receptor. So the receptor is a body structure that will detect any changes or deviation from the set point and send input in the form of nerve impulses to the control center. So the receptor will detect the changes and send the impulse to the control center. The control center will evaluate the input and it will generate output commands where they are needed. So the control center will evaluate the input, and then after that they will send commands to the effector. The effector will receive input from the control center and produces response or the effect that changes the control condition. So the effector, for example muscles or gland, they will produce the response that will correct the stimulus that is detected by the receptor. Subtopic 9.2, Negative feedback mechanism in controlling blood glucose. At the end of the lesson, students should be able to describe the negative feedback mechanism. Negative feedback refers to homeostatic mechanism that stop or reduce the intensity of the original stimulus and consequently causes a change in a variable that is opposite in the direction of the initial change. For example, if our body temperature increased than the normal body temperature, our body will undergo negative feedback mechanism to reduce the body temperature back to the normal level. If our body temperature is lower than the normal body temperature, our body will undergo negative feedback mechanism in order to increase the body temperature back to the normal level. So the negative feedback loops maintain a state of homeostasis or dynamic constancy of the internal environment by correcting mechanism from a set point. Example that you will learn in this chapter is blood glucose level. So in negative feedback loop, the receptor will detect the changes or the stimulus and will send the signal to the control center. The control center will compare to the set point, and then after that they will send the impulse to the effector, which are the muscles or the gland. The muscles or the gland will carry out the response where they will correct the stimulus that occur. Subtopic 9.2, Human homeostatic organ, structure and function of kidney. At the end of the lesson, students should be able to describe the structure of the nephron. Kidney is an important homeostatic organ of mammals. Function of kidney is in osmoregulation and excretion. Excretion is the elimination of waste product of metabolism from the body, while osmoregulation is the maintenance of a correct balance between water and dissolved solids in blood and tissue fluid. The kidney structure. The blood will enter each kidney via renal artery and leave via the renal vein. The kidney consists of two main regions, which are the renal cortex, the outer part here, and the inner renal medulla. Each kidney consists of millions microscopic structure called nephrons. Nephron is the functional unit of the kidney. Each nephron consists of long tubule and associated small blood vessels. A human kidney consists roughly 1 million nephrons. There are two types of nephrons. We have cortical nephrons and juxtamedullary nephron. Cortical nephron, which is this structure here, these occur mainly in the cortex. They have short loop of Henle, which extend slightly into the medulla, and they constitute about 85% in human kidney and function most importantly when the water supply is normal to excessive. So in human, the nephrons is mainly cortical nephrons. Juxtamedullary nephron, which is this structure here. These occur mainly in the medulla. The convoluted tubules occur in the cortex close to the junction with the medulla, while the long loop of Henle extend deep into the medulla and function most importantly when water is in short supply. This is the structure of the nephron. So the structure of the nephron consists of glomerulus, which is a highly coiled blood vessels. They are covered with the Bowman's capsule, and the Bowman's capsule is connected to the proximal convoluted tubule. The proximal convoluted tubule is then connected to the loop of Henle, which will then connected to the distal convoluted tubule. And distal convoluted tubule will end with the collecting duct, and the collecting duct is connected to the ureter. The nephron structure, Bowman's capsule. So Bowman's capsule is a cup shape and it is like a hollow ball that has been pressed in on one side. It consists of porous cell called podocyte that form filtration membrane, and it is involved in ultrafiltration. Its membrane permits the passage of fluid and solutes from blood in glomeruli into renal tubule but restrict the passage of blood cells and protein molecules. The glomerulus. Glomerulus is a dense network of capillaries contained in the invagination of Bowman's capsule. They are highly coiled to increase the surface area, has high number of pores to increase permeability. Proximal convoluted tubule. They are located in the cortex region of the kidney, consists of one layer of epithelial cells which are only one cell thick. They are involved in reabsorption and secretion between renal tubules and blood vessel. The proximal convoluted tubule is highly coiled to increase the surface area, and they have epithelial cell rich in mitochondria for active transport. The inner side of epithelial cells has microvilli and pinocytic vesicles. The loop of Henle. Loop of Henle consists of descending limb and ascending limb. They involve in the reabsorption of water and salt, thus enhances urine concentration process. They employ the principle of counter-current multiplier, which produces salt concentration gradient. So the descending limb is only permeable to water, while the ascending limb is only permeable to salt. Distal convoluted tubule. They are highly coiled to increase the surface area and empties its content into a collecting duct. It involved in the secretion and reabsorption process and affected by hormone called ADH and aldosterone. The last structure of the nephron, which is the collecting duct. It is a series of tubules and ducts that connect the nephrons to the ureter. It involved in the reabsorption of solute, urea, sodium chloride, and water. The epithelial cells has aquaporine channels that can allow water to cross the epithelial cells, and they are affected by ADH. This is the end for this lesson. See you in the next video.